Hello, everybody. As Jerry mentioned, my name is Sofia Lombard. I'm a senior at Clarkson High School North who is passionate about neuroscience. This past summer, I joined Dr. Rudolph Tanzi's lab at Massachusetts General Hospital as a research intern. And ever since, I've been working with various members of Dr. Tanzi's lab on my own research project regarding the role of AQP4 in the lymphatic system and Alzheimer's disease. And so due to these interests, when Jerry invited me to moderate the Open Box Science seminar today, I was very thrilled. And I'm excited to introduce our speaker, Dr. Darshan Sapkota, who will be presenting on the neurobiological significance of alternative modes of mRNA translation in astrocytes. He is the first author of an excellent paper published last year titled AQP4 Stop Codon Readthrough Facilitates Amyloid Clearance from the Brain. Just a little bit of background information. Dr. Sapkota received his PhD in developmental neurobiology from SUNY Buffalo and completed his postdoctoral training in molecular neuroscience with Joseph Doherty at WashU in St. Louis. Currently, Dr. Sapkota is a brand new PI at the University of Texas at Dallas, where he's currently studying the regulation of mRNA translation in brain function and disease. Finally, one fun fact about Dr. Sapkota is that he loves music as much as he loves neuroscience. So thank you for being here today, Dr. Sapkota, and we'd love for you to take it away.
Thank you, Sophia. Okay, so Zeri, let's share my screen, and I'm seeing the same option now. So if you could do what you did 5 minutes ago, and let me share my slide. That'll be great. Okay. There you go. There that. All right, so as Sophia mentioned, I started here not long ago. And so I'm in the Department of Biological Sciences and the Department of Neuroscience at UT Dallas. And we study these non-canonical or alternative modes of mRNA translation, both in neurons and astrocytes, essentially asking what is the neurobiological significance of proteins arising from these modes of translation. And so for those that don't know what I'm talking about when I say alternative modes of translation, here is this cartoon. And so,
sorry to interrupt. Still a presenter mode. Would you mind to swap the displays?
Okay. So let's see. So for that, I have to. Oh, there you go. Swap display. Good now. Am I good now, Zeri?
Not yet.
You still okay? Let's see.
Yeah, perfect. Thank you.
Did he say?
Yeah, great. Thanks.
Okay, very cool. Sorry about the interruption. Okay. So this cartoon shows the conventional or typical modes of translation, which we see in our textbook. So we have this cartoon of mRNA. The two ribosomal subunits come and they start translation at the end of the 5' UTR. And down there at the end of the coding sequence, we've got this stop codon where the two ribosomal subunits disassemble and you get this protein that corresponds to the coding sequence. That's normal translation. But it turns out there are a few transcripts for weeds. Some of these translating ribosomes, the ADS complexes, disregard the stop codon and continue into the 3' UTR, giving rise to the C-terminally extended protein. The phenomenon is called as stop codon read-through. And furthermore, for some transcripts, the ribosomes can start translation from within the 5' UTR. That's called as upstream initiation. And then still for others, the ribosomes can initiate from within the coding sequence. That is called as downstream initiation. And so these upstream and downstream initiation events give rise to this N-terminally extended or truncated protein variants, as you see. And so I use the term non-canonical translation to encompass these events, okay? And so these events were described per individual transcripts, but then at the genomic level, how prevalent they were in the brain wasn't that well known. And so I wanted to tackle that knowledge gap as a postdoc in Joe Daugherty's lab. And so I started by developing this approach that I call TRAP-RF that allows us to detect the read-through. And so TRAP stands for Translating Ribosome Affinity Purification, and RF for ribosome footprinting. In TRAP, we use transgenic mice that express GFP tag ribosomes. So RPL10A is one of the ribosomal proteins. And so we express that GFP tag ribosomes under either neuron-specific promoter SNAP25 or astrocyte-specific promoter ALDH1L1, or we could use microglia-specific promoter and so on. And so essentially the idea is to express these GFP tag ribosomes in these specific cell types. And so I homogenize the brains from these transgenic mice. And as you can imagine now, either neuronal or astrocyte-specific mRNAs were studied with GFP tagged ribosomes. And so I could use magnetic beads coupled to anti-GFP antibody and selectively pull down GFP tagged mRNA, ribosomes along with mRNAs. And so TRAP is basically a way to purify mRNA from specific cell types. And next I subjected this mRNA to ribosome footprinting, where I degraded that mRNA using RNAs. And now, wherever ribosomes were bound along the mRNA, those regions were protected from that enzymatic degradation. And so I purified those ribosome-protected fragments that we call ribosome footprints, and I prepared the library and deep sequenced the libraries. And so now these footprints, as you see, they give me positional information. That is to say, where ribosomes are present along the mRNA body. And now, when I map these reads back to the mouse transcriptome, most of the transcripts were like map2, where all the reads map to the coding sequence indicated by this blue line, because that's where ribosomes are. But then there were these few transcripts, including AQP4, for which some of the reads map to the 3' UTR, suggesting there were ribosomes there. And so now this is what led me speculate if transcripts like AQP4 were undergoing read-through. And so in total, there were 50 transcripts, some neuronal, some astrocyte-specific, the rest shared between the two cell types that showed at least 1% read-through. When I say 1%, at least 1% of the translating ribosomes going into the 3' UTR. And so, having identified these read-through candidates, I developed a luciferase assay to confirm this event using this orthogonal approach, which I'm not going to describe in the interest of time. And so essentially I validated a bunch of these 50 candidates using this reporter assay, okay? And so with that done, now I wanted to ask what read-through does functionally. Now that you have this extended protein, and given that translation is very energy expensive process, it shouldn't be happening for just random no reason. there should be a function. And so what does it do? And so for that, I focused on aquaporin 4. And I use that term AQP4X to indicate that read-through extended variant, okay, X for extended. And so this cartoon here shows that AQP4 is expressed along the astrocyte cell membrane, but it's particularly concentrated at these astrocyte processes called as NFET that wrap the blood vessels in the brain. And now, if I buy a commercial aquaporin 4 antibody, it's going to recognize both the normal length AQP4 and the extended AQP4. And so now I developed this antibody, anti-AQP4X, using the read-through peptide as the epitope. And then when I did immunofluorescence staining using these two antibodies, this is what I saw. The commercial antibody gave a signal everywhere in the brain. The signal was primarily perivascular, but it was also present outside the blood vessels, kind of consistent with that cartoon that we saw there. But then the read-through specific antibody gave me a signal that was exclusively perivascular, suggesting that stop codon read-through somehow was responsible for docking this AQP4 to the NFET, right? So this was exciting, this specific localization signal coming out of stop codon read-through. And so continuing to ask functional question, I took 3 approaches. First, I developed this mouse, generated this mouse, which I call AQP4-NOX. What I did was I added 2 extra stop codons behind the annotated stop codon to prevent the ribosomes from going into the 3' UTR. And so, as indicated in this immunostaining panel, wild type shows both perivascular and non-perivascular staining with anti-AQP4 antibody. But then in NOX, the perivascular staining is gone, but the non-perivascular diffuse signal is still present, okay? So that's one approach, a loss of function approach, mouse genetics approach. Second approach, I generated this mouse, which I call AQP4 all X, where I mutated the stop codon to a sense codon to allow 100% of the ribosomes to go into the 3' UTR. And as indicated by this Western blot, I do see this increased production of AQP4X in the all-X mouse compared to the wild-type mouse. So it's a gain-of-function approach. And a third approach, I screened thousands of drugs using my luciferase assay, remember, that I developed to validate read-through. And I identified a few drugs that could enhance stop codon read-through. So it's a pharmacological manipulation of read-through. And so using these reagents, I did a lot of experiments in Joe's lab, and I'm continuing to do so in my own lab now. And so now I want to show you some of the data focusing on Alzheimer's disease. And so here, what I did was I took an AD mouse model and I crossed it to my AQP4-NOX mouse. And what I see is the level of soluble amyloid-β is significantly increased in the absence of AQP4X, suggesting that you need this stop codon read-through of AQP4 for this efficient removal of A-beta from the brain. Our complementary approach, here is pharmacological experiment. In blue is a drug that enhances read-through. And when I administer this drug into this AD mouse brain, it does result in this decrease in the soluble amyloid-β compared to this negative control drug T, or this vehicle control. And so enhancing read-through does enhance A-beta clearance from the brain. And so mechanistically, we are still working on that aspect, but we think that it's basically by modulating the Glimpatic system that AQP4X might be removing amyloid-β from the brain. Okay, so that's about our key data focusing on Alzheimer's disease. And so we have also shown the role of AQP4 read-through in this second disease called as Neuromyelitis optica. And so this is an autoimmune disease, and it's like multiple sclerosis, but it primarily impacts the optic nerve and the spinal cord. And back in 2004, it was shown that the antiserum harvested from neuromyelitis optica patients react primarily perivascularly, okay? So known in 2004. And then it was known that aquaporin 4 was the autoantigen in this disease, neuromyelitis optica. But then given that the antiserum primarily binds perivascularly, we hypothesized maybe read-through AQP4 is the real autoantigen. And to test that, a graduate student in the lab, Hamza Lalami, he did this immunoprecipitation experiment. So Michael Levi, who is a neurologist at Harvard, he shared serum samples collected from NMO patients or healthy population. And then we did immunoprecipitation using those serum samples and using brain lysates from these mice, wild type, no X. Remember, no X has no read-through AQP4, but it still has normal AQP4. And then lysate from all X, which has this overexpression of AQP4X, right? And so as you see, healthy serum, as expected, doesn't pull down anything. But then NMO serum sample, it does pull down aquaporin 4 from the wild type lysate. But then that binding is abolished in the NOx mouse, you see, which still has the normal length AQP4. And so this suggests that it is AQP4X that is the autoantigen. And consistent with that idea, this all-X lysate, which has a lot of AQP4X, it does show this increased reactivity, okay? And so we saw that the read-through version is the real autoantigen here. So I think it's a completely new lens to look at this disease now, okay? Okay, so I think that's all I have, in terms of stop codon read-through, the key data. Of course, we have additional data, but I will stop there and then I will circle back to this cartoon. And so stop codon read-through is one of the mechanisms of non-canonical translation. What about upstream and downstream initiation sites? And so to identify those initiation sites, again, I did ribosome footprinting experiments, but I did this in neuron glia culture with and without KCl treatment. Potassium chloride, as you know, activates neurons. And so with this paradigm, now I was able to have this extra layer of information. That is to say, does neuronal activity modulate upstream and downstream initiation sites or not? And so I subjected this neuron glia culture to this lysis in presence of this drug, homoharintonin. Now, homoharintonin is a drug that binds to initiating ribosomes, okay? So ribosomes that have just incorporated that first amino acid, methionine or whatever, and it arrests those ribosomes, but allows all the elongating ribosomes to run off the transcript. And so what you are capturing is only initiating ribosomes. So again, I did ribosome footprinting experiment and prepared these ribosome footprints, prepared the libraries. And when I mapped the libraries back to the transcriptome, here is the pattern that I see, an example, GRINA, which is a transcript that codes for NMDA receptor subunit. And so 0 here is the annotated AUZ, initiation site. And you see that huge peak showing that I'm really capturing initiating ribosomes. But in addition to that, there is this upstream peak at -39 position, in the 5' UTR, suggesting that this transcript might be undergoing this upstream initiation event and potentially forming this N-terminally extended protein. So that's how I identified upstream and downstream initiation sites, okay? We validated the peptides or protein products coming out of those non-canonical initiation sites using mass spectrometry. And so this does confirm our ribosome footprinting experiment. So this is just a list. We are not going to enumerate all those transcripts. This is just to show that an orthogonal approach does confirm our ribosome footprinting findings. Now that I had this, with and without KCL approach, I wanted to ask, does neuronal activity regulate the uses of initiation sites? And so in fact, that's the case. So this heat map shows that in response to KCL, as you see in the first column here, most of the transcripts show increased uses of annotated initiation site, which is inconsistent with the idea that you have this de novo protein synthesis in response to neuronal activity. And that is accompanied by this decrease in the uses of upstream and downstream initiation sites, you see, in blue. But then there were a few transcripts that showed the opposite trend, you see, decreased uses of annotated initiation site and a corresponding increase in the uses of upstream initiation sites or downstream initiation sites. So the take-home message here is that neuronal activity results in this de novo protein synthesis, but it also regulates the choice of different initiation sites, okay? And by the way, some of those upstream and downstream initiation sites are in frame with respect to that annotated AUZ, and others are out of frame, suggesting that you might be making totally different peptides in response to activity, okay? So that's the message there. And I think I will wrap up my story about upstream and downstream initiation sites here, and just to summarize key points. And so the approach we developed, TRAP-RF, it allows us to profile translation in specific cell types in the brain. And so I think it's a cool technique to, to probe mRNA translation in a cell type of your choice. A second point, a key point here is that this specific brain transcripts, so stop codon read-through. Now, when I say specific, the point here is to emphasize that it's not a genome-wide noise. It's a very particular subset of transcripts that are undergoing read-through, suggesting that the phenomenon is regulated. It's not a random phenomenon. And as a matter of fact, we have found some cis-regulatory elements and trans factors that might be regulating read-through in the brain, okay? So that's one, a second key point here. And I hope I have convinced you that the read-through version of AQP4 is the perivascular version, and it's a potential therapeutic target for Alzheimer's disease and neuromyelitis optica. And then non-canonical initiation, as I showed, it's regulated by neuronal activity, probably by other things in the brain. And so really the take-home message here is that, from our research is that these novel N-terminal and C-terminal protein variants, I think these are really new ways to look into brain function and disease, because we are really talking about protein variants that the world doesn't know exist, And so what do these proteins do in the brain? And so that's what drives our research in our lab here. And we are also interested in the mechanism that regulates these, N-terminal, read-through and upstream and downstream initiation events. And so with that, I'll stop there. And I hope I have delivered this message that, what we read in textbooks, about mRNA translation, that knowledge is not complete. There are these, underappreciated mechanisms, generating highly functional proteins in the brain, okay? And with that, thanks a lot for your attention.
Thank you, Dr. Sapkota, for that wonderful presentation. I'd like to open the chat for any questions, so feel free to type directly in the chat, or you can use the raise the hand feature on your Zoom platforms.
Let's see. Oh, do I have to stop sharing in order to go to the chat box? I use Microsoft Teams here a lot. I used to use Zoom. Okay. Very cool.
There is a question in the chat from Felipe asking you to please elaborate on your hypothesized mechanism by which upregulation of AQP4X enhances glymphatic clearance of amyloid-β. What functions are gained by increased AQP4X expression, and what does the X do? Are there any potential drawbacks of AQP4X overexpression?
Okay, so that's a cool question. And so, yeah, I could have used a cartoon of the Glimpatic system, but the Glimpatic system posits that, we have this subarachnoid space, which is filled with the cerebrospinal fluid, and then the arteries that are penetrating the brain, they have this, what is called as Perco-Robin space, the periarterial space, which is in continuum with the subarachnoid space. And so the idea is that the cerebrospinal fluid enters the periarterial space. And from the periarterial space, the cerebrospinal fluid goes into the brain parenchyma. And from there, the CSF goes to the perivenous space. And from the perivenous space, it goes to the deep cervical lymph node, among other things. And so the idea is that the CSF is traveling in that way, you see, from the subarachnoid space to the periarterial region, to the brain, to the perivenous space, to the lymph node. And so as the CSF travels in that way, it clears away metabolic waste products, that are present in the brain parenchyma. And it is said that it is hypothesized that the astrocyte and feed localized AQP4 somehow promotes that passage, that journey of the cerebrospinal fluid, because aquaporin-4 is a water channel, probably by regulating water transportation in the brain. It somehow gives the CSF that direction, okay? And then by giving that CSF its direction, AQP4X might be promoting that glymphatic flow. And so that's a hypothesis. And so we are testing that hypothesis using the systema magna dye injection and this, the assay for the glymphatic system. And we are using our all-X and no-X mice in that assay. And so we will come up with solid answers sometime in the coming summer. But that's the idea. Second part of the question, what is the potential downside of having a lot of AQP4X? We have that all-X mouse, which has a lot of AQP4X. One thing I will tell you is that all-X mouse, it overexpresses AQP4X, but surprisingly, we saw that the perivascular localization of AQP4 is somehow perturbed in that all-X mouse, which suggests that you need normal length AQP4 as well, but this, perfect localization of AQP4 at the end feed. So that could be one potential downside. We don't know what it means in terms of neuromyelitis optica pathology. We are pursuing a hypothesis there that is, crossing our all-X mice to neuromyelitis optica disease models and see if the disease pathology becomes worse or better. So that is still in the pipeline. I see a second question in the chat box here. You mentioned that commercial antibodies detect both AQP4 and AQP4X. Have you found any ways to differentiate the two under fluorescent staining? Or is it the primary difference just the location of the different forms? Okay, so how do we, in AQP4 no-X mice, which don't have the elongated version, we see only non-perivascular diffuse localization of AQP4. And so that does confirm that, it is only the read-through version that goes to the perivascular needs, okay? It is the normal length AQP4 that is away from the perivascular region. And so that's one point, one piece of data that tells us that the two AQP4 variants are differentially localized. But at this time, we have that antibody, anti-AQP4X, that selectively recognizes the elongated version. There is no way to have an antibody that only recognizes the normal length version. And so, but I hope that mouse genetics data, no-X data, I hope that convinced you that the perivascular AQP4 is gone in that mouse. So the normal length AQP4 is away from the perivascular space. Okay.
We have a question from Zung Fu Li. So go ahead.
Yes, can you hear me?
Yeah.
Oh, yes, thank you, Professor, for the talk. So for your final part of the talk, you mentioned that non-canonical initiations could be regulated by the neural activation. So I just wondered, since it's activity dependent, did you really dive into the different neural types, such as excitatory neuron? or inhibitory neurons, whether these different types of neurons activation would contribute to different effects of the initiation?
Yeah, school question. Unfortunately, my neuron glia mixed culture. It was just, just pan neuronal preparation. We had just, all sorts of neurons there. And so I really don't have an answer to your question, but that's a very cool question. So one thing I'm gonna do is, right now we are developing these antibodies to selectively recognize those peptide variants, coming out of upstream or downstream events. And so maybe by doing immunofluorescence staining, in the mouse brain, let's say I could use, neuronal stimulation paradigms, PTZ, that kind of paradigm, and see where those non-canonical proteins are present, which cell types are expressing that. Another approach would be to do that trap RF, the technique that I used to detect stop codon read-through in vivo, a trap RF, but unfortunately, it was almost impossible to do the detection of this, upstream and downstream events in vivo, because you have to treat the brain with that drug, homoharingtonin. You want to capture that, those initiating ribosomes. And so there is this time factor. And so I spent a few months trying to do this, trap approach to detect cell type specific alternative initiation events. So the experiment didn't work. So now I'm left with this approach, have these antibodies and see which cell types express various variants. Yeah. And what does it mean functionally? Yeah. We'll go from there. Yeah. Very long road, unfortunately. Yeah.
Thank you.
Yeah.
I have a quick question as well. What biomarkers would be necessary to establish the clinical significance of compounds that help modulate AQP4 read-through, as you mentioned a few of them?
Yeah, okay. Never thought about that. What biomarkers, would be needed to assay the efficiency of these read-through modulating compounds? Okay, so I'll tell you this: the compounds that we identified are not specific to AQP4. They enhance read-through for all other transcripts. And so there is that downside. That hampers the potential clinical use of those compounds. Say, for example, in Alzheimer's disease, the perivascular localization of AQP4 is perturbed. It's reduced. And so it would be, a cool idea to try to restore that perivascular pool of AQP4 pharmacologically. But at least our compounds, they are not specific towards, any transcripts. And so they are gonna result in this potential, off-target effects. And so in terms of biomarkers, I would say, rather than a specific biomarker, I would say if we want to test the efficiency of those compounds, we should be doing ELISA or Western blot and try to see how we are modulating a read-through, various bands, the normal length band and slightly, higher molecular weight band, the read-through band. How are we modulating those two proteins? I think that's the way to go, Sofia.
Thank you for that answer. There's a follow-up question in the chat from Felipe. How does the X component promote perivascular localization? You mentioned the need for regular isoforms of AQP4 for proper localization. So may you please elaborate on the interaction between the different isoforms and perhaps other proteins as well?
Sure. So one thing we did was this, protein-protein interaction, assay for that, and we found that AQP4X interacts with certain scaffolding proteins present at the blood-brain barrier, including alpha-syntrophin, which is known to tether AQP4 to the NFET needs, okay? And so that's the mechanism. That is to say, the read-through peptide interacts with that tethering protein, the scaffolding protein. That's the mechanism. And so your second part there, why is it that you also need the regular normal length AQP4? So you see, AQP4 in physiology doesn't exist as monomers, okay? Yeah, AQP4 first, it forms these tetramers, 4 protein molecules coming together, forming tetramers. And then these tetramers again come together, dozens and dozens of tetramers come together and form what is called as supramolecular complexes or orthogonal array particles. So you can see those huge particles under electron microscopy. And so one possibility here is that even though it is the read-through peptide that interacts with that scaffolding protein, the AQP4X variant might still be interacting with the normal length AQP4 variant, in terms of forming this supramolecular complexes. And so, in those complexes, I think both the versions of AQP4 are present. And so that's one hypothesis that we have. And we do have data supporting this hypothesis. I just didn't include those data. I thought we had 2 speakers today, and so I wanted to, keep my talk within that half an hour time span. But yeah, I hope I answered your question. Protein-protein interaction, and then AQP4 interacting with normal AQP4 and forming OAPs, orthogonal array particles. Yeah.
Sorry, I do have one more follow-up question. So what does your research tell us regarding the role of the lymphatic system and water channel regulation in Alzheimer's disease specifically?
So I think our research really provides a whole new genetic mechanism to explain the lymphatic system. When the system was discovered in 2012 by Meakin-Niedergard, she used her lab from the University of Rochester. Her lab used global knockout, global aquaporin-4 knockout mouse model, and then in global knockout, Meakin-Niedergard groups, they found this decrease in perivascular, the lymphatic clearance, and then they hypothesized that it is the perivascular AQP4 that was modulating the system. But really, they were, using the global knockout, they were not really differentiating the role of perivascular versus non-perivascular AQP4. And so now, by identifying this, unknown mechanism, the stop codon read-through, and identifying this, C-terminally elongated version, we really provide this, a genetic handle to manipulate the lymphatic system. That is to say, our NOX mouse model is the true loss of function model for the lymphatic system, because it still has non-perivascular AQP4, has this, selective absence of non-perivascular version. And so that's the true genetic model. And Conversely, AQP4 all-X mouse, it's a new gain-of-function mouse model for the glymphatic system. And so again, as I mentioned, we are assaying the glymphatic system in these mouse models. And we are really excited about some of the preliminary data we have. And so back to your question, I think our research really provides a new, genetic mechanism to explain the glymphatic system.
Thank you for that insightful answer. If anyone else has a question, please feel free to use the chat or the raise the hand feature. We'd be happy to get to your question.
Just a quick question. Are you looking at any gene read-through in microglia too?
So I have a few read-through candidates. I'm developing antibodies. And so as I mentioned, Zeri, before the seminar began, I'm from Nepal. And so I have a friend back in Nepal. He has this biotech company and he's very clever. What he does is he immunizes goats and then gives the goats to these poor farmers in the Himalayan mountain region. And then so he doesn't have to raise goats, you see. Farmers are raising the goats and goats are producing antibodies. And then when the goats are ready for a meat harvest, he goes and he collects the blood and gives the meat to farmer, And so it's a win-win situation to produce cheap antibodies, you see, goat antibodies. And so that's how I'm producing, a lot of antibodies. And so some of the candidates that I'm focusing on, they are expressed in microglia. One of them is PLAT, plasminogen activator tissue type. It's expressed ubiquitously in the brain. So what I'm trying to say is I'm focusing on transcripts, read-through transcripts that are both cell type specific and ubiquitous. We'll see, in terms of microglia specific function. Yeah, you have to see. But yeah, so there are, microglial transcripts that's so read through. Yeah. Okay. Doesn't look like we have further questions. So thanks a lot, everybody. Thanks for your attention. Thanks for coming here. If you are, interested in any, if you saw any interesting candidate in my list there, read through list, feel free to reach out to me. I have open arms if you want to collaborate. Okay. Otherwise, we'll run into one another at conferences or, other places along our journey. Yeah.
Thank you so much,
Thank
Dr.
you.
Sapkota. It's a wonderful presentation.